I just want to thank the first two speakers because that was actually a pretty amazing lead-in to what I'm going to be talking about. Both a little bit of electron transfer and some protein interactions with these class of molecules known as conjugated oligoelectrolytes, which for the entirety of the talk I'll refer to as COEs. I always like to start with this slide just in part because it's pretty, but also in part because it sort of illustrates the diversity of organisms out in the environment. And you can see the color change here, which is associated with just temperature tolerance, but these are all phototrophic organisms at the Grand Prismatic Spring in Yellowstone, so kind of a nice little view. But I guess first I have to thank my collaborators and my PI, who unfortunately couldn't be here today. He was one of the people that got stuck with all the weather. But also my colleagues Chelsea and Hengjing are in the audience, and they'll be giving posters. So be sure to check them out. And I have to thank my funding agency, the Institute for Collaborative Biotechnologies, for letting me explore this work. Also, maybe some of you know Huiji. She's still around. And Logan Garner was one of the original researchers that sort of founded this project and worked and synthesized many of the conjugated oligoelectrolytes that we work with nowadays. So a quick outline. I'll be talking a little bit about some of the founding experiments with conjugated oligoelectrolytes and the fact that the mechanism of their action has been rather elusive over the years. So this work was started back in 2010, and we've been trying to figure out why these molecules are helping. And then a quick bit of context with Schuonella and just sort of an energetic landscape in terms of thinking about how these molecules might be working. And then 3 hypotheses that I sort of directed my experiments around and how I eventually tested them. So first of all, I have to introduce you to this class of molecules. Conjugated oligoelectrolytes bear a pi delocalized hydrophobic core, which is semiconducting and fluorescent. And there are polar pendant groups that impart water solubility. What's important about these molecules, though, is that their amphiphilic structure mimics the amphiphilic structure of the lipid bilayer. And so they will spontaneously intercalate into the membrane from an aqueous environment. So they'll partition into the membrane with a pretty strong affinity. Now this is powerful because presumably we're now introducing a semiconducting moiety into a rather insulating structure. So if we move forward, some of the original experiments that we saw were just simple yeast-based MFCs, where we saw when we added these molecules, we saw a boost in voltage output and current output. And it's just a simple U-tube type fuel cell device, abiotic cathode. Abiotic cathode. And then in future experiments, we actually reversed the process and shown that we can inject charge into Schuonella using these molecules as well. So if you track current over time, the moment when we add these conjugated oligoelectrolytes, depending on concentration, we see a large current draw from the cathodic biofilm, which is rather impressive. So the Schuonella is actually turning over fumarate into succinate, and if you track the electron equivalence in the current, so if you integrate this, you get this solid trace here, and then if you, with high-performance liquid chromatography, if you track succinate production, you actually see that it tracks very well with the current that we integrate. So we do know that it's quantitatively transferring electrons to the terminal acceptor for the bacterium. So this was kind of a novel and important experiment, but we still didn't understand what was going on. So there is no mechanistic information here, and so what I'm going to talk about today a little bit is some of my work where I've been monitoring current output, just chronoamperometry, and I've done a lot of cyclic voltammetry and some DPV, but I won't talk about the DPV today. If you want to talk about that, I can go into it maybe in person. A little bit of microscopy, so I actually quantified the biofilm, and I just used a simple single-chamber 3-electrode type device to probe these sorts of things. So we've heard a good bit about these, but you poise the potential between your reference electrode and your working electrode, and that allows you to just monitor what's happening at the anode. Quick but a bit of biotic context. I've set this energy landscape up in terms of silver chloride just for convenience, but you could honestly set it up against anything. Okay, yeah, so for many humans and other animals in nature, we're consuming our food and using the reducing power of oxygen, the oxidizing power of oxygen to power our metabolism. And that provides sort of a voltage that powers our bodies. Green plants are taking sunlight and splitting water and exciting electrons to an excited state, which then serves as the electron donor for the rest of their metabolism. When we think about S.oneidensis, it resides in a much more narrow regime, but it still is consuming, say, lactate, going through this MTR pathway that the previous speaker talked about, utilizing flavins, and respiring on a variety of minerals in nature. And so the potential of these various minerals kind of determines how much energy you can get out, which is what Cesar was talking about. What's really important about S.oneidensis, though, is you can utilize a working electrode as a proxy for any of these minerals. And so if we set up an electrode at, say, 200 millivolts, we see contributions from both the MTR pathway and flavins. And this is sort of the punchline. When we add COEs, you see more current. But I'll get into exactly what I mean by that. But what's really important about S.oneidensis is that it does have these two mechanisms, and they show up independently of each other in voltammetry. So if you look at the flavin-mediated electron transfer, it resides at a potential a little bit lower than the direct electron transfer. And so we actually see conjugated oligoelectrolytes boosting some electron transfer there as well. So the question really that I'm going to get into is how COEs interact with this system. The first hypothesis is simply that COEs increase anodic current, which occurs rapidly, and because of this increased anodic respiration, coulombic efficiency is also increasing. The second hypothesis is that it's due to increased respiration and not something like toxicity, and then also that the increased respiration promotes electrode colonization, and perhaps that coulombic efficiency is increasing on a per cell basis, so each cell is getting better at producing electricity. And then thirdly, COEs are accelerating direct electron transfer rather than mediated electron transfer. But that does have implications for mediated electron transfer because these are interacting pathways, as Katerina talked about. Flavins are actually directly interacting with cytochromes. So here's a more detailed schematic view of the device I used. If we zoom in on the biofilm, really just see that lactate is being stoichiometrically converted to acetate, and it's actually producing 4 electrons per lactate molecule, ideally. So if we look at an established biofilm of S.oneidensis, it sort of produces a steady state current that you can measure, and if I take an SEM at, say, the end of operation, so I go through here and monitor current output, and at these breaks in the current I've interrogated electrochemically, but at the end of operation I chemically fix it with, say, formaldehyde and then dehydrate it and look at it under scanning electron microscopy, and I actually go in by hand and count these things on the picture. You can sort of get a quantitative view of how many cells there are. But then when I add DSSN or another conjugated oligoelectrolyte to the system, I see a rapid boost in the current output. And if you zoom in even farther, you can really see that it occurs in a matter of minutes, seconds, which is compared to the doubling time of a S.oneidensis culture in minimal media, which is upwards of 90 minutes. So we're actually seeing electron transfer accelerate almost instantaneously, and then it stays sustained over many hours. And if I continue to, if I change the media or anything like that, this current stays elevated. So this is indicative of transmembrane electron transport being accelerated and not something like growth, say, instantaneously. However, when I do take an SEM of the fixed biofilm afterwards, I see a dramatic increase in the quantity of cells in the electrode, which is indicative that respiration is increasing. It is allowing the cells to reproduce more quickly or reproduce at all. So going back to some of these hypotheses, some of these data are consistent, at least, with these initial hypotheses that anodic current is increased and it happens rapidly. It's due to respiration, and that indeed does promote colonization. Now, I alluded to this earlier, every lactate molecule should produce 4 electrons if everything's working right, if you have 100% Coulombic efficiency. But as we all know, in bacterial systems, it's impossible to get 100% Coulombic efficiency. So if we integrate the charge from between these 2 points, we can see that with COEs added, you get a larger amount of charge collected than without COEs. The lactate consumption by the biofilm ends up being somewhat similar, but it's a little bit elevated in the COE-containing culture. So if we crunch these numbers a little bit, we can see that without conjugated oligoelectrolytes, the Coulombic efficiency as compared to what you would expect, so that's the ideal value. So if we take this amount of lactate and convert it to an ideal amount of charge using the 1:4 ratio, we can see that we get about a 51% Coulombic efficiency, which is respectable. But when we add conjugated oligoelectrolytes, the Coulombic efficiency is actually going way up. So we're seeing more current, and it's more efficient at doing it. And so if you look at the ratio between these two things, it's about 1.7, so it's a 70% increase in the Coulombic efficiency. But that's compared to the number of cells on the electrode has increased by 3.1-fold. So if we actually compare these numbers, it doesn't seem that the cells are getting better on a per cell basis. So that's kind of interesting. It seems like the bulk system is getting better, but it's not because each individual cell is getting better at transferring charge. So if we go back to this, we have indeed improved Coulombic efficiency, but it's not happening on a per cell basis. That doesn't seem to be correlated with the number of cells. So I'm going to zoom in a little bit further into the membrane, which thankfully Katerina talked about as well. So we have the MTR protein complex. I left out OMCA just for clarity, but these proteins can undergo direct electron transfer with an electrode, or they can undergo mediated electron transfer and interact with these flavin derivatives that we saw earlier. And that's all coming out of the quinone pool purportedly. So this is just a reminder of the current output I was showing. And at these 3 points, I interrogated it electrochemically and did cyclic voltammetry. So as you would expect, the current increases from here to here to here, and indeed we see current increase from 1 to 2 to 3 as well. And these are all poised at 200 millivolts, which is what I showed earlier in the energy landscape diagram. Importantly, if we take a this is the flavin, the mediated electron transfer, and this is the direct electron transfer regime. And importantly, if we take a derivative of these traces, you can actually see that we have the flavin, and then there's actually a flavin semiquinone, so that's a bound flavin state that shows up. And then the direct electron transfer peak is sort of broad out here. And then if we go in and look at the trace where conjugated oligoelectrolytes have been added, so they're presumably sticking themselves in the membrane like this and somehow interacting with these proteins. If we interrogate them electrochemically the same way, you can see that the flavin regime of the electron transfer is relatively unchanged, and the direct electron transfer has been boosted a great deal. So again, if we take a derivative of this, you see the flavin, you see the flavin semiquinone peak, and the direct electron transfer peak has been dramatically accelerated. So what we see from this is that these molecules actually seem to be boosting the innate machinery in the cell and not necessarily acting like a molecular wire or something like that. So earlier I talked about how these molecules might possibly act like a molecular wire because they have this semiconducting moiety through the middle. It would make sense for them to be a conductor through the membrane, but it doesn't actually seem that that's how they're working. They're actually making the cell better at its own business. So I overlaid the two plots just for visual comparison. You can really see that these two devices prior to adding the COE were operating very identically. And these are all done at 5 millivolts per second. But when conjugated oligoelectrolytes were added, the direct electron transfer dramatically increases, and the flavin electron transfer is relatively unchanged, although you do see a little bit of an acceleration, which is presumably because this is increased, and these are interacting pathways. So that's the conclusion of it, and what I have shown is that direct electron transfer definitely is increasing, and that does seem to accelerate mediated electron transfer as well. So that's all I have for you today. So you're asking about metal groups on the COE?
Yeah. So you said it shows is that a methyl group or a metal?
Oh, it's methyl, yeah. Methyl, yeah. Trimethyl ammonium iodide is the solubilizing group. But so.
Do you have any abiotic controls?
Yeah, I have done some abiotic controls, and these molecules really do not contribute to current in any way. It's basically inert at the electrode, especially at 200 millivolts. So if you actually, well, if you do cyclic voltammetry to characterize the HOMO and LUMO of these, you see that the highest occupied molecular orbital would be probably out here somewhere. Oh, this plot looks silly. Yeah, yeah. I hope they didn't all look like that. Okay, good. Yeah, if you did cyclic voltammetry of the COE, you'd see it would have a redox peak out here somewhere. So it's not being oxidized or anything.
Kind of following up with that, have you looked at these COEs in cells that do not themselves have any electron transfer?
I have, and we have actually done a lot of different work with that. So we've done experiments with yeast and E. coli, which both are not very good extracellular electron transport organisms, and we do see that it increases EET, but it seems to be limited sort of by the cell's innate ability to do it. So it's not anything near what you would see in terms of current here. It does boost it. So some of those original experiments that I showed, it's maybe a 5-fold increase, but it's still a very low current. Okay.
Nice talk. I wonder, an increase in Coulombic efficiency, I always find that something interesting. In a way, what else is happening then? If you have more cells, you should have a lower Coulombic efficiency. And so they inversely correlate in this case.
Right.
So can you speculate on what is actually happening that increases the Coulombic efficiency?
Sure, and I agree that is a little bit puzzling. So in the literature, there are some reports that the direct electron transfer pathway is much more efficient than the mediated electron transfer. In part because, in the reactor I'm using, these flavins don't necessarily stay interacting with the cells, so they can diffuse off, especially if they're reduced. You then sort of lose those reducing equivalents.
But that would lead to a same Coulombic efficiency with these mediators. They would get reoxidized over time. It would be a kinetic thing.
Right.
So would it be possible like that in your efficiency, for example, that rather than making current, you make hydrogen gas? And have you measured the hydrogen? Because normally
that actually is possible.
Lactate, it makes acidated hydrogen gas. It comes out.
Right. Yeah, and
So maybe it came out, and now by having more direct electron transfer, you don't have hydrogen.
that is definitely a possibility. So he was, for anybody that didn't hear it, he was saying maybe hydrogen is off-gassing, and that's because of the direct electron transfer being more predominant. You're not generating as much hydrogen, which would be lost. That's a pretty reasonable explanation for it, but I have not measured the hydrogen.
Last question, Mike?
Yeah, can you say that they are replacing your COEs, that they are replacing your kinones or anything else in the electron transport chain? Because I would think that if they are some kind of shortcut, that they would also shortcut the proton transport mechanism and then would shortcut the breathing, respiration.
Right. So his question was whether the COEs might act as a replacement for quinones or something like that. As far as I know, the COEs are not redox active. So it's more, if anything, electrons would be tunneling through them, but they can't accept an electron or donate an electron without degrading. So I don't think that's what's going on.
Okay, let's thank our speaker again.